Let's begin the last unit of the course, Unit 22, the digestive system. Here we will discuss Part A, general principles, Part B, anatomy, and Part C, digestion. Under digestion, we'll look at what is digestion, we'll discuss chemical digestion, then regulation, and finally absorption. The digestive system carries out two processes, as we mentioned when we introduced with the donut: digestion and absorption. Digestion is the breakdown of large insoluble food particles into small soluble food particles. There are two kinds of digestion. One is mechanical breakdown, and the other is chemical breakdown. Absorption is the transport of digested food from the lumen of the GI tract, that's the space inside, into the body. So you remember the hole in the donut is technically outside of the body. Absorption brings the nutrients into the body. What is mechanical breakdown? Well, just like it sounds, it includes chewing, mastication. It includes various types of motility, movement, such as mixing and propulsion in the GI tract. For example, peristalsis in the entire gut. This is a wave-like contraction of the wall of the GI tract, propelling the material along. Turning in the stomach. Segmentation in the small intestine. Chemical breakdown, also called hydrolysis. You remember it's a type of reaction in which you add water to split a molecule. It uses enzymes to split large molecules into small molecules. Small molecules are the products of digestion. Enzymes are secreted by cells and glands evolved in luminal or cavital digestion. Enzymes may be bound to the intestinal wall, to the membranes. These are referred to as brush border enzymes. They're involved in membrane or contact digestion. Chemical digestion of carbohydrates, lipids, and proteins will be summarized. You're not responsible for many of the finer details. Let's look at peristalsis in a little more detail. In the tube, circular muscle will contract. The tube will constrict, and a wave of constriction moves along the tube, repelling the contents of the tube to the other end. Let's look at carbohydrate digestion. You see a polysaccharide. It uses carbohydrases to split into disaccharides. That's a molecule with two sugar units. You may remember from the unit on matter. That's further split by carbohydrases into monosaccharides. So the monosaccharides are the product of carbohydrate digestion. It's accomplished mainly by carbohydrases in the saliva, such as salivary amylase, intestinal juice, such as maltase. In the pancreatic juice, there's more amylase, pancreatic amylase. It produces 80% glucose. So most of the results of carbohydrate digestion consists of glucose. There is fructose and galactose. Fructose is changed to glucose in the intestinal epithelial cells. Galactose is changed to glucose in the liver cells. So as you'll see, the end result is in the blood, carbohydrates are in the form practically of 100% glucose. Almost all the carbohydrates in the blood are glucose. Lipid digestion. We'll use triglycerides as an example, or so are other lipids as well. Triglycerides are digested by various lipases into monoglycerides and fatty acids. The monoglyceride consists of the glycerol portion and the fatty acid. Two fatty acids are split from the triglyceride. This is accomplished mainly by lipases in the pancreatic juice, but there are lipases in gastric juice and intestinal juice. Other lipids include cholesterol, phospholipids, glycerol. What about protein digestion? Protein digestion involves proteins being split into polypeptides, which are then split into individual amino acids. This is accomplished mainly by proteases. In the gastric juice, pepsin is a common form. Another common form is trypsin, found in the pancreatic juice. Now, there are many other proteases as well. Other proteases include enterokinase. Proteases are secreted in an inactive state. They're called zymogens and activated after secretion. Now, you can see why that would be the case. If they were active inside the cells, they would begin to dissolve the cells. Protein digestion requires hydrochloric acid to split large proteins. Hydrochloric acid is produced by the cells of the stomach. It activates pepsinogen to pepsin. Pepsinogen is an inactive form of that enzyme. It also serves to kill microorganisms, so it's one of the body's defenses, part of the immune system. Hydrochloric acid is neutralized by bicarbonate ions in the small intestine. Now, what about the content of the GI tract? Well, in the mouth, food is formed into a ball for swallowing. This is the bolus. In the stomach, the bolus mixes with liquid and becomes semi-liquid material called chyme. Chyme passes into the small intestine where most of the digestion occurs. The undigested wastes become feces in the large intestine. This table will summarize chemical digestion. It's divided into 5 columns. First, lists the structure and the general function of that structure. The second lists the secretions that are found within that structure, the pH of those secretions, and approximately the milliliters per day that are produced. The site of production is listed in the 3rd column. The 4th column shows the chemical content of the secretions. Now, they all include water and mucus, which won't be repeated. The functions are listed in the 4th column. Let's consider the mouth. The mouth is involved mainly in mechanical breakdown. It does contain saliva, pH of about 6, roughly 1,500 milliliters a day are secreted. Saliva is produced by the salivary glands. Among the chemicals in the saliva is salivary amylase, which breaks down starch to maltose. Second structure is the esophagus. Now, the esophagus is simply a connection between the mouth and the stomach. Third component is the stomach. This has 2 general functions. One is storage, and the other is digestion. It contains gastric juice. It's highly acidic, a pH of about 1.5, and roughly 2,000 milliliters a day are secreted. The gastric juice is secreted by cells in the stomach wall. Among the chemical contents would be proteases, such as pepsin, and hydrochloric acid. Proteases begin the splitting of proteins to polypeptides. The hydrochloric acid splits large proteins. It activates something called pepsinogen, and it kills microorganisms. About 3,500 milliliters total is secreted into the stomach. The next structure is the small intestine. It carries out digestion and absorption. It's probably the most important component in digesting and absorbing the food materials. Roughly 8,500 milliliters a day is absorbed. The secretions in the small intestine include intestinal juice, pH of about 7.6, roughly 1,500 milliliters a day is secreted. The juice is produced by cells in the intestinal wall. It contains maltase, enterokinase, also lipases, amylase, sucrase, lactase, and a variety of others. The maltase splits maltose to glucose, and the enterokinase activates trypsinogen. Also operating in the small intestine is pancreatic juice. Pancreatic juice has a pH of about 8. Again, roughly 1,500 milliliters a day is secreted. Pancreas produces the pancreatic juice. It contains pancreatic amylase and bicarbonate ions. It also contains proteases such as trypsin and lipase. It functions to break starch into maltose, neutralize acids, break down polypeptides into amino acids, and begins to digest fats to fatty acids. Also found in the small intestine is bile. It has a pH of about 8.2, roughly 500 milliliters a day is secreted. It's produced by the liver. It contains bile salts and bicarbonate ions. It helps to emulsify fats, more about that later, and neutralize the acids that were produced in the stomach. The volume secreted is something like 3,500 milliliters a day. And then there's the large intestine. The large intestine concentrates feces and stores wastes. About 350 milliliters a day of material is absorbed. Its functions are varied. There's no digestive function, but it does absorb water and ions. It contains bacteria that help digest cellulose. They also produce certain vitamins like K and B12, thiamine, and riboflavin. The bacteria also produce gases. That's the flatulence that's produced. Feces are about 3/4 water and 1/4 solid material. Of the solids, about 30% are dead bacteria, 30% are undigested roughage, and 40% organic and inorganic material. There are more bacteria in the large intestine than there are cells in the human body. The volume secreted, 0. We've mentioned secretions. We said they include mucus and water secreted by all subdivisions of the GI tract. The average amount exiting the GI tract per day by absorption and excretion is 9,000 milliliters a day. 8,500 milliliters a day is absorbed from the small intestine. From the large intestine, 350 milliliters a day is absorbed. 150 milliliters per day is excreted. Total, 9,000 milliliters a day is absorbed from the GI tract. Where does the material come from that's absorbed? Is it all the material that you've swallowed? No. Ingestion brings in 2,000 milliliters a day. The average of all secretions entering the GI tract is roughly 7,000 milliliters a day. Saliva, 1,500. Gastric juice, 2,000. Intestinal juice, 1,500. Pancreatic juice, 1,500. Bile, 500. Now let's look at regulation of digestive tract function. Regulation involves both endocrine and neural control. And two processes are involved: control of motility, that's the chewing, the peristalsis, and so forth, and secretion. Endocrine control, remember, involves hormones acting on target tissues. I know you've seen this diagram many times, but it doesn't hurt to bring it up again. Stimulus acts on a gland, the gland secretes the hormone acts on a target, and the target takes an action. The endocrine gland cells may be in accessory organs or within the wall of the GI tract. The target organs include parts of the GI tract and the accessory organs. So you see, it may begin to get kind of complicated. Let's enlarge the wall of the GI tract. That's it in yellow. Nervous control involves two different reflexes. One is a central nervous system reflex. It's also called a long or vasovagal reflex. I'll call it the CNS, central nervous reflex. It involves sensory transmission by way of sensory neurons to the central nervous system, and motor transmission from the central nervous system back to the site of action. This is controlled by the parasympathetic nervous system, mainly the vagus nerve. On the other hand, local nervous system control, it's also referred to as short or myenteric reflexes, but I'll call it local nervous system reflexes, involve sensory and motor transmission within the wall of the GI tract. Now notice the difference. The CNS control sends transmission to the central nervous system and back. The local control transmits within the wall. The impulses don't leave the alimentary canal. The stimulus acts on receptors. These may be in external sense organs like taste and smell, or they may be located in the wall of the GI tract. You see one sensory receptor as part of the central reflex and one part of the local reflex. This table will summarize regulation of the processes in the GI tract. It's divided into three columns. The first column lists the organ, the second the action, and the third lists the control of that action. Let's start with the mouth. The mouth involves motility. It's voluntary through the central nervous system. About the salivary glands. Their action increases saliva secretion, and it's under control of central reflexes by way of sight, smell, and taste, and so forth. So when you smell dinner cooking, you begin to salivate. The esophagus is the tube, as we said, to the stomach. It involves increased motility, which occurs by way of central and local reflexes. About stomach regulation. Stomach involves increased motility and secretion, as well as decreased motility. There are three phases. The cephalic phase involves the central reflex by way of sight, smell, and taste of food. This causes you to begin to salivate. Gastric phase involves both central and local reflexes when the stomach wall is stretched. Also, there's a hormone called gastrin from the stomach wall when the wall is stretched or when proteins are present. And then there's the intestinal phase. It involves both central and local control. Stretch of the intestinal wall slows gastric emptying. This is called the enterogastric reflex, and it allows time for the stomach to do a little more digesting of its food. You remember endocrine control. We keep coming back to that 5-part scheme. Keeping that in mind at this point is pretty useful in trying to figure out hormonal control in the digestive system. Consider the hormone gastrin. What's the gland that secretes gastrin? Well, it's the stomach wall, at least specific cells in the stomach wall. What's the stimulus that acts on the gland when the wall is stretched or proteins are present? What about the organ that the hormone acts on? What's the target of the hormone? The target is the stomach. More specifically, cells that carry out secretion and smooth muscle cells that carry out motility. Finally, what's the action? Well, we just mentioned it. Increased motility and secretion. Let's summarize and add a few more details. Stomach control involves the cranial and cervical parts of the spinal cord. The cephalic phase, sensory input from sight and taste and so forth, increases stomach activity. In the gastric phase, sensory input from stretch of the stomach wall by both CNS and LNS reflexes and gastrin and histamine increase stomach activity. So histamine is also a hormone in the stomach. Intestinal phase, the role for this is to slow stomach activity. Sensory input comes from stretch of the intestinal wall by CNS reflex activity. The vagus input to the stomach is inhibited. Sympathetic input to the stomach slows activity. Hormone secretin and cholecystokinin slow stomach activity. This is the enterogastric reflex. And again, the role is to slow down the activity of the stomach, which would move the material onto the small intestine to give it time to be digested. Let's look at regulation of the small intestine. It involves increased motility and secretion, and it's under control of both central and local nervous system reflexes when the stomach wall is stretched. And then there's the pancreas. Pancreas involves an increased secretion of enzymes as well as an increased secretion of bicarbonate ions. The enzyme secretion is under central control in the cephalic and gastric phases of stomach action. There's also a hormone called cholecystokinin from the intestinal wall when proteins and lipids are present. The bicarbonate secretion tends to neutralize the acids. This is under control of another hormone called secretin. This is from the intestinal wall when acid is present in the small intestine. You might wonder, why is it called secretin? Well, because it was found a number of years ago that when blood supply of 2 cats was connected and one cat was fed food, the pancreas of the other cat secreted pancreatic juice. So they thought, it must be a signal transmitted by the blood from one cat to the other. And because it caused secretion, they named it secretin. You might also note that there's no local control of the pancreas because, remember, local control involves the wall of the intestine, and the pancreas is not part of that wall. And then finally, we have the gallbladder. The gallbladder will increase the release of bile. Remember, it's produced by the liver. This is accomplished by central reflexes due to stretch of the intestinal wall, as well as the hormone cholecystokinin from intestinal wall cells when proteins and lipids are present in the small intestine. And finally, there's control of the large intestine. The large intestine involves mainly motility because not a lot of juices are secreted into the large intestine. Control is both central control when food is present in the stomach, called the gastrocholic reflex, also central control when chyme reaches the duodenum. This is the duodenocolic reflex. Then both central and local control occurs when feces are in the rectum. This is called the defecation reflex. How often have you sat down for a fancy meal only to have your youngest child announce that he or she has to go to the bathroom? He's not being difficult. It's just biology, the gastrocholic reflex. Defecation involves several steps. It's controlled through the lumbar and sacral parts of the spinal cord. Feces in the rectum stretch the rectum. Sensory nerve impulses transmit information to the lower part of the spinal cord. Motor impulses then are transmitted by way of the parasympathetic nervous system to the rectum to stimulate contraction of the rectum. Inhibitory nerve impulses, meanwhile, also relax the internal anal sphincter. Voluntary nerve impulses from the brain keep the external anal sphincter contracted until defecation occurs. Absorption is the transport of digested food from the lumen of the GI tract into the body. Most absorption occurs in the small intestine, into the villi, these microscopic finger-like processes. It requires increased internal surface area. We describe that under anatomy. The macroscopic surface area of the small intestine is something like 2.5 square meters. That is, if you slit open the small intestine and laid it out flat, the internal surface would encompass that area. Microscopic surface area, on the other hand, is something like 400 square meters. So it's a huge increase in surface area. Absorption involves both active and passive processes. After absorption, all nutrients are transported to the liver. Carbohydrates and proteins are transported by the hepatic portal system. Lipids are transported mainly by the lymphatic system as well as the cardiovascular system. Active processes of absorption include active transport. Sodium, for example, calcium ions, and many other ions are actively transported. Carrier-mediated transport is involved in the transport of most monosaccharides. Glucose is particularly important, and amino acids. Glucose, fructose, and galactose are absorbed into the intestinal cell walls. Most of the fructose and galactose are changed into glucose in those cells. Lipid absorption, on the other hand, is more complex. Let's look at lipid digestion and absorption. This is a view of the small intestine, an intestinal cell wall, highly enlarged, and a lymphatic capillary. Various stages of fat digestion are fat, illustrated with the F, fatty acids, and monoglycerides. Micelles are microscopic spheres of phospholipids that spontaneously form around the fatty acids and the monoglycerides. Chylomicrons are transport vesicles in the intestinal cells. Let's look at the details of lipid digestion and absorption. The first step is emulsification. It's a physical breakdown of large lipid drops into small lipid drops. Bile salts are used for this process. The next step is the fats are digested. Remember, this is hydrolysis, and lipases are used. The fats are broken down into fatty acids and monoglycerides. Absorption into the intestinal wall then takes place. The bile salts form micelles. The micelles surround the fatty acids and the monoglycerides, and the fatty acids and monoglycerides diffuse from the micelles into the intestinal cell wall. The micelles have come in contact with the intestinal wall and allows this to happen. Empty micelles then move down the small intestine and are absorbed through the wall of the ileum and recycled to the liver. This is called the enterohepatic circulation. However, this is not the end of the story. Once inside the intestinal cells, the fatty acids and the monoglycerides are reassembled into fat. The fats are packaged into these transport vesicles called chylomicrons, and the chylomicrons are released from the epithelial cells by exocytosis into the lymphatic system. They are then transported to the liver, mostly by the lymphatic and cardiovascular systems. Some are transported directly by the blood in the hepatic portal system of the liver. Passive processes of absorption includes osmosis, the absorption of water, diffusion of some ions, such as potassium and chloride ions. Electrolyte is often lost in diarrhea. Potassium. Remember, the net movement of a substance by diffusion is from its high to its low concentration. So normally potassium is absorbed out of the intestine. Why potassium? In diarrhea, excess water is present in the intestines. What is the relationship between passive absorption of this ion and the increased concentration of water in the intestines? Increased water concentration during diarrhea decreases the potassium concentration, so it reverses the concentration gradient. The gradient is now from the outside to the inside. The result is the net diffusion of potassium from the outside of the intestine, where the concentration is high, to the inside, where the concentration is lower. And so the potassium is lost. Well, now we're ready to progress to the next part of Unit 22, the digestive system. This would be metabolism.